Malaria.
What is it?
Why do we care?
How do we stop it?
Malaria is a disease prominent in third world countries caused by the Plasmodium parasite. The Plasmodium life cycle exists in two distinct stages, one inside and one outside the human body. The goal of this project is to develop a drug to attack the Plasmodium parasite while still inside the body.
Current antimalarial compounds are prone to failure due to the rapid mutation of the parasite which builds up a resistance. Our drug attempts to alleviate this problem as it focuses more on the red blood cell as opposed to the parasite itself. The group of compounds that we're interested in are called the triazolopyrazines. We use these because of their size and structure, which enables them to lock onto the reactor side of a red blood cell, which causes an increase in the sodium ion concentration within the cell, killing the parasite inside.
Pyrazine is a known organic compound that acts well as an antimalarial drug. What we've been trying to do in our experiments is modify two specific reactor sites on a pyrazine core to try and maximize its efficiency. We do this in 3 stages. First, we add an aromatic aldehyde to the pyrazine core. This is one of our functional groups that we're trying to modify. Next is oxidative cyclization. This is a bit complicated, but basically we're just trying to change the shape of this structure to get the pyrazine core that we want. After that, we finally add an alcohol, and this is the second functional group that we're interested in.
So this is the first step in the formation of the pyrazine molecule that we're trying to get, where we're undergoing a condensation reaction to deposit this aromatic aldehyde onto our pyrazine. In doing so, we're going to create a small molecule, in this case water, that's going to get ejected out in the process of the reaction, and we're also depositing our aromatic aldehyde onto our pyrazine.
In experimental chemistry, we never know whether the reaction we've done has completed, and we find this out using TLC. It's thin layer chromatography, bro.
Thin layer chromatography, or TLC, is an analytical form of chemistry that allows us to separate our mixture into its varying components based on their polarity. We have a thin silicate base layer that has both our reactants and products on it. We then place this in a solvent of predetermined polarity, which, as it adhedes up the silicate surface, will drag the compounds within these mixtures to varying heights depending on their compositions. We can use TLC for 2 different things: checking whether our reaction has gone to completion, or checking for the purity of our mixture when we compare it with replicate TLCs.
All of the stages of our project are examples of different processes in a field of research known as pharmaceutical chemistry. We, along with scientists from all over the world, collectively post our findings on an online database so that we may collaboratively work towards our goal. This is known as open source drug discovery. Oh, hey guys!